Hello everyone, good morning. It's a bit rainy weather here in Uppsala and
Recording in progress.
by the way, I don't know how the students are distributed across Sweden, or they are mostly in Uppsala or Stockholm.
It's national. We
Okay,
got even national students.
okay, that's great. That's great. So it's quite diverse. Fantastic. Uh, so the title that I thought will be relevant is single-cell RNA-seq data analysis, and I have a subtitle, a very short introduction, because this is in a way a compressed MP3 version of a workshop that we have at MBIS. My colleagues Osa and Roy and Susan has spent a lot of time preparing this course, and I try to summarize it and shorten it into 1 hour. So it's a lot to go through. The longest story short, I will be scratching the surface only and just going through the standard workflow. And at the end, I will introduce few resources which I hope you will find useful to expand your knowledge in this type of data analysis. So when we are studying biology, we try to understand how an organ works in order to link it to a phenotype or disease that we are studying or investigating. And one way to do that is profiling the transcriptome or expression of genes across different tissues. And we can do that by different means. We can either do bulk RNA-seq, where it's like a soup of different cells, and we try to explore the expression profile. Or we can go to single-cell resolution, where we specifically look into individual cells to understand their expression differences. We can also look at the expression spatially with spatial transcriptomic technologies. Then, in addition to the expression profile, you can also map at which region of tissue these are expressed. Few points about pros and cons of each of these techniques. In bulk, what we get is a cell average expression. It is cheap and easy, and you can develop data or generate data from many replicates. But it comes with its drawbacks as well. It is very heterogeneous, the signals might be diluted, and you do not have any spatial information. In single-cell RNA-seq, you can go from bulk to single-cell level resolution, and you can characterize new cell types. It also gives you information about cell states. However, it is expensive, it has tedious library prep, and again, similar to bulk, it does not give you spatial information. On the other hand, we have spatial transcriptomics, which from the name, it's explanatory. We have spatial resolution. The resolution can be at spot level or cell level. So in the spot level, you do not have single cell resolution. Still, you have collection of cells within a given spot. But you have the spatial resolution. Similar to single cell RNA-seq, it is expensive. You have tedious library prep, and you have custom set. I mean, you cannot characterize the full transcriptome if you are, for example, using Xenium. But with Visium, you can go for the full transcriptome. And in that case, you have a spot resolution. One of the my bad. So one of the key differences between bulk RNA-seq and single-cell RNA-seq is the expression profile that you see and the differences among the samples. So in bulk RNA-seq, you have a good correlation. Of course, it depends which set we are looking, but overall, in general, the overall expression is well correlated when you're looking into replicates from the same group. However, in single-cell RNA-seq, there are could be major differences between cells. You have dropout events. For example, in this case, you have genes expressed in one cell but not expressed in another cell. You have over dispersion. The expression is showing a high variation between different cells. And also you could have outliers or genes that have very high expression. So for this reason, we have to have a thorough evaluation or a correctional normalization, which we will talk a bit further in coming slides. We can have the data from single-cell RNA-seq in two formats. It's either full length or it is tag-based. So in the full length, similar to bulk RNA-seq, we can characterize a full transcriptome and a full transcript. It is usually the case for the plate-based technologies, and it gives you an even coverage of transcript. At the same time, it helps you to detect splice variants, while in tag-based, you target for only 3' or 5' ends, and it has limitation in detection of splice variants. Now, let me tell you very quickly about the library preparation and sequencing, and it's very simplified version of that. There are more details in it, but it starts with cell isolation. And then you have reverse transcription. It is where we add UMI. I will talk about UMI in a second. And then after reverse transcription, we have amplification of the cDNA, and then eventually tagmentation and library construction. So during duplicate amplification, we generate many copies of identical fragments from the original molecule. So it's important to distinguish the duplicates, and we address that by using unique molecular identifier, or UMI. It can help to quantify the original molecule. These sequences are 4 to 8 base pairs long, and they are random barcodes. They are added before amplification step, and here is one example. You have cell barcode, and then in addition to that, you have the UMI. So UMI, as you can see, is different for different molecules. For example, if you take HCTP as an example, as you can see, the UMI is similar. And in theory, these two coming from the same molecule, so we can collapse them. And if you look at ARL1, there are two reads with two different UMIs representing two independent copies of the molecule. These will help us when we are going to do the downstream analysis, because we demultiplex and then we keep the unique UMIs. There are different sequencing protocols. I'm not gonna bore you by going through each of these cells in this table, but to highlight main point is that you will have a microfluidic strategy, well plate base, and fluidigm, and last but not least, split pool. So I recently learned about the split pool myself. So the microfluid is where you generate the droplets, and then you encapsulate the cells within the droplets together with the beads and barcodes. And in well plate base, from the name you can guess, you have wells on the plate, and the cells are distributed in these wells. There are different names for different library preps and sequencing results, but all of them have UMIs, and you can target for full length or tagmentation. One of the key differences between these technologies are the throughput. So it can go from few hundreds to thousands and even millions of cells. The split pool, if I can say only one thing about the split pool, is that you do not need a lot of hardware compared to the other technologies. It makes it a bit cheaper. And I have mostly worked with 10x genomics chromium data, and today the dataset that you're going to work with is coming from this technology. So I will very quickly go through the analysis workflow, which starts with the raw reads. You are mostly based on the answers that you gave. I think you're more familiar with FASTQ files. You can check the quality of the raw reads by FastQC, and the first step of the analysis is to map the raw reads to the reference genome. And if you have 10x data, you use CellRanger, which gives you a summary HTML. We will look into that, and a BAM file, which is a binary file of the mapped reads, and CellRanger molecular info file, which is HDF5 file, and that's what you load in by scMPy. The raw read step, you do not need to worry about that. It's already done, and what you get is the HDF5 files. As I told you, you use the scMPy, and the pre-processing and the following steps, everything will be done with the scMPy, or tools which have been implemented in the scMPy. The first step after you load your data is QC. And you generate N features, mitochondrial genes percentage, and so on, to check the quality of the data. And then you filter out the low quality cells and genes and check for the proportion of doublets. I will talk about doublets in a few next few slides. You can also check for the cell cycle status of the cell, because sometimes you may need to account for cell cycle effect in your data analysis. The single cell RNA-seq is high dimensional data set. So you in a way try to control for that by doing a feature selection and identify highly variable genes. Now, after this step, you move to the next step, which is visualization, and there you start with data integration. Here there are different methods. The method that you use, all of them try to correct for technical variation you have in the samples that you try to analyze. And here you would use harmony. In addition to that, after this step, you do dimensionality reduction with the scMPy. You generate PCA and UMAP to visualize the data and see the distribution of the cells in reduced dimensional space. Last but not least is the downstream analysis, and it starts with clustering. With this step, you try to label cells showing different expression profile. And you use this data, this information to do differential expression analysis. And this is the comparison between clusters or group. There are, of course, different and additional analysis that you can do, like trajectory analysis, gene set enrichment analysis, and so on. But it doesn't fit in this slot that I have, and also it makes the practical much lengthy. So with this, I would like to start walking you through the QC steps. This is one report that you get from CellRanger. It is a QC report, and it gives you important information, like the estimated number of cells, the mean reads per cells, and median gene per cell. So there is some extra information that you can read in terms of like the quality of the reads and bases, and in addition to that, you have the mapping. So it shows that the proportion of the reads that have been confidently mapped to genome, it is very low, 21%. Do you have any idea what can be the problem here? Okay, someone said bad quality.
You're not raising the good reference, maybe?
Yeah, that's actually that. And so you can already see that in this report. So if you look at the sample information, first of all, the chemistry shows that it is 3' tagmentation. And also the reference, it is chromosome 21. The transcriptome is chromosome 21. So it's only one of the chromosomes that we have mapped the data. So that's important pieces of information that you can already see within this report. The question is that how could it tell that there are introns included? So because it has the full reference, and it can also have the information of intronic regions. So we have different isoforms, and some of the reads may align into intronic region as well. Good questions. And another pieces of information that we can extract from the QC report is the barcode rank plot. I will dive into that further, but very short version of that is that it gives us information about proportion of empty droplets. I will talk about that in a few seconds. So all the analysis that we do can be done with different tools depending on your environment of interest. You can either use R, which you use SORAT. The problem there is that there's a limitation in number of cells that you can load. I have heard that with SORAT version 5 you can go up to 1 million, but I haven't tested myself. But an alternative is to use scMPy in Python, which can apparently handle many more cells. So there are different modules in scMPy. The first one is read and write, and you have preprocessing, which you can check the quality of the data, generate some QC metrics, and also filter the cells and genes, and scaling the data. In scMPy tools, you generate PCA, UMAP, and also do clustering. And last but not least, with plot module, you plot the data or results that you have generated. I explained this here because just to know that these small abbreviation that you see, PP, TL, and PL, is referring to different modules in scMPy. So the data is like this. You have a matrix. It's a cell, a count matrix with dimension is number of cells and number of genes, by number of genes. And in addition to that, you have QC metrics, latent variables that need to be saved in an object or in a format. And how to save this is if you're using SORAT, you have SORAT object with different slots. And if you're using scMPy, it is and data format. And you have the X matrix, which you see here. You have number of observation, which are cells, and then number of variables, which are genes. And in each of these axes, you can have extended information in different slots. So for example, in where you have the metadata variables and multi-dimensional calculations like produce dimensionalities like PCA, TSNE, and so on. And last but not least, you have unstructured data, which is the data which you cannot fit in relation to any of these dimensions, any of these axes. For example, you can save the variation ratio of the PCs in this slot. And data is also popular formats for spatial transcriptomics data as well. So very quickly, what you get from raw data is you do the QC, the multiplexing, mapping, and quantification. And the quantification results in a matrix that then you have cells in rows and genes on column. It is a sparse matrix, and you have only non-zero elements stored in the matrix. Going back to a note that we discussed very quickly is the tools that develop to parse technical reads content are quite diverse. But if you're working with 10x data, CellRanger is the tool to go. And always remember to make sure to have the right genome version and annotation version, because it can influence the readout, especially if you're going to compare to other results. So it's important pieces of information that you need to consider in the data analysis. So empty droplets and barcode rank plot. In this plot, you have in the x-axis you have the barcode ordered, and then you have the UMI counts. So in the droplets technique, you have droplets which encapsulate the cells, And there should be a balance between how many cells you load. If you load few cells, then you end up with many empty droplets. What you are looking is into, you should expect cliff and knee patterns, which is like this, that you see in this plot. So the idea here is to see a drop in the distribution, or in the line. So that shows that all of a sudden you have many empty droplets. That's the spot that Cell Ranger, using an algorithm called similar to empty drops, try to identify this breakpoint. And what you get at the end is the filtered set, and these are the cells or droplets that have cells, and the empty droplets have been excluded. And if you load too many cells, you end up in a situation that you have more than one cell in each of these droplets. And it could be homotypic when you have the same cell types, or could be heterotypic when you have cells from different cell types. There are different approaches to detect these doublets by using high number of reads and features that you identify in your dataset, or you can make a comparison with the simulated doublets. Here is an example of the simulated doublets. So you have your own cells distributed in the reduced dimensional space in PCs, and then you generate artificial droplets or artificial points, and then you visualize it again. And what you see, if you see a cluster of the cells which are going together with the artificial doublets, you mark them as doublets. So how do you know that what is the rate that you should expect? 10x Genomics has suggested an approximate expected multiplet rate, depending on number of cells loaded and recovered. And you can use this table in order to evaluate the picture that you see. So what you do after you generate this doublet detection is that you visualize the number of features and counts, both for the cells that have been marked as doublet and the non-labeled cells. And you can see the expression differences or the distribution differences, that the doublets usually have higher number of genes by counts. And remember to always assess the frequency and the distribution in the cells. To the left, you have t-SNE, and here you see a group of cells which shows a high doublet score. And you can already see that these cells, which are shown to look to be doublets, are grouping together, are clustered together. Sometimes you can also characterize this by making a comparison between these groups and with the rest of the clusters or groups. So if you had done some differential expression analysis, I think you would identify some cells and differentiating this group of cells. On the right-hand side, you have a UMAP. UMAP and t-SNE, I will talk about them very briefly, but it is one way of reducing the dimensionality. And here you see the distribution of the doublets, which is completely scattered. So in situations like this, where you see both the low fraction of the cells have been labeled as doublets and it is scattered across the space, you should not be worried about that. You can just continue without filtering this. But it's up to you. You can always do that. As long as you do not see them in a group or in a cluster, it should be fine to move on. The next step is filtering. We would like to get rid of cells which have low quality, and one metric that you can use is mitochondrial gene proportion or fraction. And there you mark those little cells that have high expression of the mitochondrial genes, which can be indicative of poor sample quality, leading to a high fraction of apoptotic or lysine cells. In tumor biopsies, you may also have the situation that this picture may have increased due to metabolic activity or necrosis. In addition to this filtering, there might be still some genes like MALAT1, which pops up all the time in single-cell RNA-seq data, and it's important to remove those for differential gene expression analysis. And last but not least, we exclude cells which do not provide enough information, like those cells that do not have many genes expressed. Cell cycle is also another metric that you can look into, because sometimes what you see may not be due to true differences between cells, but can be affected by cell cycle states. And you can calculate the states by states of the cell in the cell cycle by using a gene marker list, which is already implemented in ScanPy and SOROD, and it gives you a score. Remember that if you're interested in studying cell cycle, you do not need to correct for cell cycle scores or labels. And it is very important to note that SOROD and ScanPy use a fixed cutoff. So don't just go with the labeling. Just go, instead of labels that you receive from these tools, look at the scores. The normalization is a very important part of this workflow, because what we are interested in is to capture the biological variation, in particular cell types. But there are technical variation as well, which we may not have direct control on those, and we can control that by other means, which I will come back to that when we talk about data integration. But all in all, in normalization, the idea is to make the expression comparable across samples, cells, and genes. And we do that by scaling, transforming, and removal of unwanted variation. The scaling is probably a simplified version of the classification, is either by going for sequencing depth, which is simply generating a size factor and controlling for the total depth. And in addition to that, you can also control or normalize or scale for gene length. Remember, if you're working with the tag data sets like 3' or 5', correction for gene length is meaningless. In addition to that, there are tools like zero inflated normalization that you can use in order to control or normalize by dropout rate. And scaling results in relative abundance differences and expression among genes, which that helps us to characterize the differences between the expression between the cells. I'm not going to walk you through all of these different methods implemented in different tools, but one take-home message here is that all of the methods control for sequencing depth, and today you will use log normalization implemented in ScanPy. Transformation is also important because it helps you to adjust the distribution of expression variance to better capture biological variation and reduce the impact of technical effects. And log transformation is popular in bulk RNA, helps you to mimic a normal distribution, and in single cell it results in negative binomial distribution. The next point is z-score or z-score transformation. It is a linear transformation. You try to scale it in a way to have a mean of 0 and a standard deviation of 1, which is also called centering and scaling. And PCA is sensitive to highly expressed genes with high variance. And so scaling like this for each gene helps to have a better performance of PCA. Of course, it's an immediate question which method to use. In most cases, it is enough to do depth normalization. It suffices for most of the cases, and log transformation. While we control for the technical variation, you can correct for confounding factors. There are different tools, different methods that you use. You can already pass given metrics like mitochondrial read percentage or cell cycle to regress out when you do the normalization. And remember that normalization does not influence the clustering, but can have a strong impact on differential expression. Now I switch the gears to talk about feature selection. Single-cell RNA-seq, as I told you, is a high-dimensional data, and to reduce the feature dimension and redundancy, we do feature selection, which helps us to avoid overfitting in downstream analysis. After filtering and normalization, there might still be non-informative genes with zero count expression, and we should also remove those. Remember that some of the genes can be tissue specific and can be informative to distinguish cell types. We should not make a very strong filtering on these. And in SCAMPA, you can calculate highly variable genes by this function. So what's the picture that I want you to have in mind is that when the average expression increases, we usually have the tendency of having high dispersion or variation as well. So by controlling for these, we can first of all, by normalization, we can control for these, and then we select the highly variable genes that can provide some information for downstream analysis. We will go to dimensionality reduction soon, and that's the place we will use these genes. And here you can see that these has been marked as black dots. In data set that we have high dimensionality, we have the curse of dimensionality where, for example, we have thousands of genes for given cells. And it's important to make sure that we look at an informative dimension of the data. And you can use different approaches, like linear approaches or nonlinear approaches. We will talk about PCA from linear approach and t-SNE and UMAP from nonlinear. I'm not going to walk you into every points here. There are different mathematical reasoning and background about those, which personally I don't know all of those by heart either. But what we want to do is to identify PCs or principal components that explain a larger fraction of the variance, and those will be used for other approaches. Remember that single cell is a nonlinear data set, so PC may not fit well, and tools or approaches like t-SNE or UMAP can serve better. These two approaches are graph-based, and they use PCs, for example, and they can capture local structure. UMAPs not only captures the local but also global structure. And here is one of the key differences between these two approaches, because t-SNE can preserve the similarity between closely related cells but not the global structure. That's in contrast what UMAP does. And it helps to have better scalability and interpretable. It's more interpretable compared to t-SNE. And this is the output that you get. So on PC, you have on principal components figure, you see the samples. There is a large overlap between them. But when you look at t-SNE and UMAP, you have a better distribution of the cells. And some of you have worked with single-cell RNA-seq data. What's your thought when you look at these UMAP, for example? Is there any problem with it, or it looks fantastic? Harmonization, clustering. So if I rephrase these, so COVID-17 is a separate cluster. It's bad. So that's interesting observation. Which you will see that in your practical as well. That's true. I take this as if you disregard whether this sample is has a bad quality or not. I don't call it bad. It is different. But what you see as a whole is that you see the samples, you can distinguish samples in this reduced dimensional space. So still there could be some overlap between them, but we may have batch effect. That's the point that I'm trying to make, is that when we do not control for the batch effect, we may treat these samples showing very different expression profile. But in data integration, we can see whether it has a different picture or not. Thanks, it was an interesting points that you highlighted. And I would like here to finish the dimensionality reduction by saying 2 notes. Remember to check the parameters in any of these reduced dimensionality approaches that you use, like percentage of mitochondria, etc. And check whether these information can influence the picture that you see. Is there any bias rooting from those factors or not? And while doing so, make sure not to overcorrect or remove confounding effects, which potentially can be related to your biological questions, and those will be important aspects of your study. So let's talk about data integration or batch correction. So this animation in a way shows the picture that you saw in the UMAP. We can see samples, we can distinguish the samples. But after integration or controlling for technical variation, we can see that we have a better overlap of the cells that can have potential overlap, potential similarity. So there is one question: how to make sure not to overcorrect? Any criteria? So in single-cell RNA-seq, you have to go a lot of times forth and back. So sometimes, for example, you have a gene of interest which have, for example, given expression profile. And if you filter those cells, those genes, so you may not see the effect when you are doing in your experiment. So that's one example that I can give you in terms of overcorrection. In technical variation, we have, for example, it can root into stress cells in different labs. We may have human errors, the library prep protocols, and etc. They can contribute the differentiation that we see, technical differentiation that we see in cases like this animation. So there are different tools that you can use. I'm not going to walk you through all of these approaches. They have been implemented in different tools like R or Python. But all of these, which can be based on correlated variables or mutual nearest neighbor, and all of these try to still preserve or maintain the biological variation while controlling for technical variation. And today you are going to use Harmony, which is an extended implementation in ScanPy. Now, after doing normalization, filtering, normalization, and dimensionality reduction, you can go with the clustering. The clock is ticking, and I have to go fast from here. But I try to make it as understandable as possible. There, in single-cell RNA-seq, this is a key step. You try to identify or label the cells showing differentiation in the reduced dimensional space. It is an unsupervised machine learning problem, which I think you are already familiar. And there are different methods. One is density-based and centroid-based and hierarchical-based approaches. Today you will use Lighten, which is an extended version of Lowine. And all of these try to, if you take this an example, all of these try to assign the data points into a centroid. So this is a k-nearest neighbor, and it calculates the distance between the points, and then it tries to see they are closer with which centroid. So in k-means, for example, you decide a number of clusters beforehand. In approaches like Lighten, you do not decide about number of clusters, but you use different resolutions. Differential expression is the next step. which you can do that with in different angles, from different angles. You either go to make a comparison between clusters, for example, cluster A versus cluster B, or you go into analyzing differences between conditions like the patient and healthy individuals, regardless of the clusters. And you will do both in the practical, and that's possible to do in rank genes group. There are different statistical methods. One is non-parametric tests, and you use Wilcoxon rank sum test, for example. Parametric tests are t-tests or t-test over estimation variance, which can be useful when you have or it's the classical case of t-test when you have a large variation in your data set, and then you use this approach. But all in all, in this small set that I tested, you see very good overlap between the different methods. What you get at the end, similar to bulk RNA-seq, list of the genes, significance level, and then the magnitude of difference between the clusters or conditions. And then you make a comparison. When you make a comparison, you can select the clusters, for example, given cluster 1 versus cluster 2, that you can see in this stacked violin plot, showing the top 5 expressed genes, differentially expressed genes between these two clusters. And in addition to that, you can make a comparison between given cluster and rest of clusters. And here you see top 25 genes in cluster 7, which has overlap with cluster 5 and 0, and which may not be very surprising, because these 3 clusters are located in this space in the vicinity of each other, so may have very similar properties. The consideration for differential expression is that the idea is to distinguish biological differences, not batch effects. So make a thorough evaluation when you are trying to make a conclusion from your data set, in order not to confuse the signals and noise. And remember to account for differences in cell numbers. In the practical, you will also correct for that, to have a fair number of cells for your differential expression, because if you have a very uneven, very large difference in number of cells, it can influence the differential expression. And choose the right statistical model. We can test few tests that I told you, and usually I use the non-parametric, and it works fine. Concluding remarks. Single-cell RNA-seq gives you an unprecedented resolution, but it comes with some challenges, like high dimensionality and sparsity. Thus, it's important to have a thorough quality control. And remember to have a proper normalization, and make sure to correct for the batch effect. And if you have cofactors, you should use more advanced statistical models for differential expression analysis. For example, scAMPy may not give you that option. You should use alternative options. And always visualize, interpret, and go forth and back. And then make sure to validate your findings. Don't go what you can identify bioinformatically. You can validate it by different means. The take-home message is that choose the right analysis tools and strategy. Be up to date in the field, which is evolving all the time. And biological insights are only as good as your data. So rigorous processing, pre-processing, modeling, and validation are crucial. Few words about the exercise. The task is to identify differences in expression profile of patients versus healthy individuals at single cell level. You have 4 patients and 4 healthy individuals, and the data has been subsampled to 1,500 cells per sample. Here, the information that you need to know is that it is a COVID-19 PBMC dataset. and there is a significant lung inflammation in elderly patients, and a lot of cells coordinate immune responses. So you want to know how large is the contribution of each cell types. This is the link, the title, and also QR code of the cells. And then one thing which I think would be worthwhile to say, if you want to get extended version of this course, go to this website, workshop single-cell RNA-seq, or apply for that, which will happen in spring. And also, last but not least, this single-cell best practices book is also fantastic. You can have a very good overview of the different methods and technical aspects and theoretical aspects of this data analysis. With this, I will be finishing here, and if you have any questions, I will be happy to answer. Almost at time, 2 minutes. Yes, please.
Thank you very much for the nice lecture and the overview of the different workflows and techniques there are. I had to leave, so I will continue here with this. Yeah, so are there any questions for Nima? Maybe I can start with a question. Where do you think will the trend of data generation in single-cell RNA sequencing go to in the next few years? You already showed some like bulk RNA sequencing, single-cell RNA sequencing, and then spatial RNA sequencing. Is bulk RNA sequencing still used, or are there still applications where you can see usage for this one, or is it everything is going to the more newer technologies?
So I think all of these technologies provide different types of information, and I mean the bulk RNA-seq is still popular. Some of the researchers still do that. It of course depends on the scientific questions, but we still receive bulk RNA-seq data. Saying that, we do see a bigger trend towards single-cell RNA-seq, because it gives you much higher resolution, and the signals that you're looking can be diluted in the bulk RNA-seq. At the same time, there are possibilities to convolute the information from the single-cell and the bulk RNA-seq. I think the beauty will be that when we can integrate different modalities. For example, we have a spatial and single-cell RNA-seq, and we try to deconvolute there as well to have the spatial information as well. I'm involved in a project that one of the potential activities or analysis will be that we combine the data or results from the spatial and single-cell.